Hello? Hello? feedback at the end of the session. And this is actually a platform given to KMCT by this company, where they are having a big for research and development manufacturing unit, so that we can integrate our subject with their subject, where we can interact and we can have a collaboration forward. So that is the actual aim of this particular event. So interact with them and make sure that we are taking the things forward with them in future. And this is on behalf of KMCT we are conducting, so this can be taken as a task for all sister concerned institution for MAC purpose.
we became completely at the mercy of the multinationals like Monsanto and all that, who could easily do this work in parallel. So, and finally we have to license money to them and pay money to them and license the technology from them. So the same thing will happen in the future too. So if you get scared and if you walk away from this and keep ourselves closed, the world is not closed. So we are all interlinked. So if you can't make something cheaper or better than others, others will do that and beat you out of the market. So this is already seen in we have seen in China and the US and other countries taking good advantage of. So what I'm trying to say in a nutshell is that all these changes are happening, but India is poised well. So one of the key things that we need to take advantage of is the growth that we have, and then focus that on the new technologies, especially in the field of biology, which we are very passionate about. You will hear in the rest of the day. But other technologies also which are important, and many of them are converging. And a very good institution like KMCTC is ideally poised because you have multiple institutions too. So most of the world has gone into silos where engineering will focus just on engineering, chemistry will focus just on chemists, and medical will focus on medical. But in the future, many of these or most of these will converge. So you need engineers who understand medicine. You need medical people who can understand engineering and work together. So the convergence is going to be key to take advantage of many other technologies. So I hope you students, especially I see a lot of women. That is a great thing to see. And so you will all take advantage of what is happening, and being here in this fantastic institution, we can all work together and try to build a better future. Thank you.
Thank you, sir. Now I request Dr. Navas KM, Chairman and Managing Trustee, KMCT Group of Institutions, to honor our chief guest, Mr. Sam Santhosh, with the Ponnada. Thank you, sir.
We are delighted to invite Dr. Ramchand C N, advisor of SaksinLifeSciencePvt Ltd, for the felicitation. Sir, please.
Good morning to everybody. Respected Mr. Dr. Navas, Mr. Sam Santhosh, Dr. Gopakumar, Manoj Kumar, Dr. Sujith, and another Manoj who is all the way came from US, and Raju, and colleagues from KMCT, the faculty, and my friends. You might have seen me maybe about a month back here. I don't know. Some of you might have been here. At that time, this hall was full. I just landed from London yesterday and straight coming here. So I did not sleep. So that's why all the sound and other problems are there. So, as I mentioned, last time when I came here, it was full. And I was actually invited earlier to give a part of a talk for a conference which was organized here. Then when I came here, slight changes, and straight away I could talk for 3 hours. I talked on NC discovery development, biology discovery development, some on nanotechnology, but then I was told by Dr. Sujith that just give some insight into entrepreneurship, and I myself is just started with the help of Sam. Sam is my mentor to become an entrepreneur, but then a lot of things I could understand the last 8-10 years. So that is what I have, the initial part of my talk at that time. And that is basically was the genesis of this interaction. So subsequent to that, I had a very good interaction with Dr. Navas for about 2 hours. We discussed quite a lot. And subsequently, he came to Cochin to some of our facility. Then we have initiated the process and what can be done. So that is what is happening right now. So what we thought that, how we can collaborate. as Sam mentioned, KMCT has several institutions apt for doing a very good research, because the resource is the most important, whether it is infrastructure, people, money, etc. So these things are there. Then what can be done? Especially if you are shifting applied research, translational research, what can be done? So certainly some kind of help from entrepreneurs or from industry is required. So that is what probably we are going to have a discussion, and you will be able to see today 5 distinct talks will be there. So Sam is going to talk on genomics or its application and what we are doing, and Raju is going to talk on KDisc, what they are doing, what we are doing currently, and Amit is going to talk on oral microbiome or role of microbiome in health and disease, and subsequently Manoj is going to talk on synthetic biology. It's a novel technology which has been, the technology was there, but it has applications coming up or came now. And I will be talking a very different subject, not last time when I was talking. It is something on nutrigenomics and reverse pharmacology and how products can be developed, which I will be talking on with my experience developing several products in that area. So we will have to discuss all those things and come to a conclusion what can be done collaboratively, especially with SciGenom and SciGenom Research Foundation. And we also conduct several conferences, in the last 10-12 years, large conferences we have done. In that also we will be able to collaborate. Now, subsequent to that, I had several interactions with Dr. Sujith, because we have been interacting, what can be done. So he mentioned that one of the facility and expertise, or at least the people available in the pharmaceutics line. In the pharmaceutics, basically, if you see, discovery part is there, which is very complex, which is not normally done in India. Discovery of the targets or the mechanism. The discovery of NCEs or the chemical entities, that's another part. That is something which is a very good chemistry understanding is there. Then the pharmaceutics side, in the formulation, there is lot can be done in that one. Because in India, most of the expertise and the development in the pharma industry is happening over there. The other area is the biopharma, which is novel biologics or biosimilars, which is not much is happening, and there is a potential area for that one. Then he also mentioned about, for example, clinical trials. India, not many clinical trials is happening, and it has never ever done a phase 1 trial, an example. But there is opportunities there. And another thing is that please remember, India has never developed a single novel chemical entity to become a global molecule. This is something, being such a big country with 26,000 pharmaceutical industries. So these are some of the things what we have to discuss and talk. So this is something we discussed over the period, and probably will continue to discuss. These are all what I mentioned is more of chemistry. Then a lot of biology will be there. That is what we are going to talk during the next 2 or 3 hours time. Then what I have also seen is that, app title was given, Bridging the Gap Between Science and Entrepreneurship. Really is the right title. Bridging the gap between science, or you can see academia, and entrepreneurship. So I'll just go back to about 15 years back, 15 or 20 years back, lot of biotechnology, bioinformatics and biopharma, lot of courses came up throughout India. And, there was one complaint, but nobody was getting a job. So what DBT did was that DBT did a big survey. At that time, so many companies wanted to get into, because I was at Vice President of Sun Pharmaceuticals. I was taking care of all the things, because I know the reality at that time, the difficulty in bringing up such a big company, such a big R&D. And the DBT has told that 90% of the people who has passed, whether BTech or MTech or MSc Biotechnology or PhD, not getting job. The same is the case with industry. Industry is telling 95% we are not getting people. What to do? It's not that, when you apply, you will get applications. But the problem is that when you interview, you realize that nobody is suitable, that skill set is not there. So this is the major problem. Neither industry is not able to hire, and the people from academy is not getting the appropriate job. What can be done for that? What I feel is that, there is a tremendous training requirement is there. So as a part of that, what we have done is that we have one Kerala State Industrial Development Corporation has supported us for 3 years. And these 3 years, they were giving the scholarship to students. And we were taking about 20 students. And we were putting them between Cochin and Chennai. So what we never trained, because I was in most of the committees in most of this university for the syllabus. And I have seen that anything and everything is there in the syllabus. But neither there is teachers, nor students, nor any of these things. Whatever we are talking, for example, synthetic biology is there in the syllabus. At least in Anna University, I know it is there. In Madras University is there, and in also SRM universities is there. But there is no one has ever done that one. So students have to study, and without appropriate training, it is not possible. So what I wanted to tell is that, so we organized a training program in which never we have gone and, trained any of this latest technology. Because I have never studied biology in my life. I've studied only maths, physics, chemistry. But over the period of time, got trained in the biology, whether it is biotechnology or natural product or synthetic chemistry, any of this thing, which is possible if you have a appropriate mentorship is there. So this is something what we have done. We trained them, these 20 students, not in advanced subjects. What we have trained is that, for example, right from IUPAC naming systems, configuration, confirmation analysis, simple synthetic chemistry, then how to make solutions, molarity, normality, equivalent to it, all those things. I can tell you, even now, 50% people fail if you make, how to make 5% glucose solution, weight by volume, volume by, nobody answers. Forget about making a buffer. So these are the problems. And without understanding this, you can't get into lab. You can't do things. You will all make mistakes. So what I wanted to tell you is that how this can be sorted out. This can be sorted out by really interacting with the industry. Or what I suggest is that everywhere I tell, put some industry people who is into research to come and train once in a month, 3 or 4 hours, and it will work. And what exactly is doing in industry? Because other thing is that, the most important thing if you talk about pharma and biopharma, what is required is that, what is regulatory, how to write patent claims, what is QC, QA, and what is the manufacturing process, what is GCP, GLP, GMP. None of these things, and this is the core of the industry that is not trained in universities. What is to be done? So these are some of the things that can be trained. So I thought that, this is probably what we will be discussing, and how we can see how we can collaborate, come out with a very solid, good plan, and so that this institute become really a well-known institute, especially in research like international institutes. I hope so. And thank you for inviting Sujith again. Thank you very much. Thanks. Thank you all.
Thank you, sir. We now cordially invite Mr. Amit Chowdhury, Head of R&D, SciGenom Labs, Kochi, for the felicitation.
Hello everyone, good morning. See very exciting faces. It reminds me of 40 years back when I was like you students. But I'm hoping that in all the talks that we have arranged, that will help you, guide you, make you read more. So again, thank you for coming. Yeah, bye.
Thank you, sir. Now I invite Dr. Manoj Kumar VP, Principal of Dental College, to deliver the vote of thanks.
our respected chief guest and resource person at today's inauguration, Mr. Sam Santhosh, who is the founder, chairman of SciGenom Labs, Dr. all the other resource persons, Dr. C N Ramchand, Dr. Amit Chaudhary, Dr. Manoj Krishan, Dr. Raju, our chairman and managing trustee, Navas sir, senior faculty members, HODs of the all the sister concerns of KMCT group of institutions, and my dear students, all the participants, very good morning to all. Today's program, I'm very immensely happy to be here with you. This program, it's a very relevant program, bridging the gap between science and entrepreneurship in life science, life care. So we have our chairman have a good vision to how to become a campus other than academia research. That is why the IEDC Innovation and Entrepreneurship Development Center is developed in 2019, that is 4 years. For the IEDC, the purpose of IEDC is to foster the culture of innovation, creativity and entrepreneurship among the students and faculty members. The main objective of IEDC is to nurture and support aspiring entrepreneurs, promote innovative thinking, and facilitate the conversion of ideas into viable business ventures. In the context of a medical campus, an IEDC plays a unique and crucial role in bridging the gap between science, healthcare, and entrepreneurship. Definitely, the Dr. Navas sir already discussed with Sam sir and associates regarding his vision. And after a long discussion, they agreed to visit here and giving you a sensitized talks on this day. So wish you all the best. And I'm directly moving on to the duty entrusted to me. First of all, I extend my sincere gratitude to chief guest and resource person, Mr. Sam Santhosh sir, to this session, and all the other resource persons, Dr. C N Ramchand, Dr. Amit Chaudhary, and Dr. Manoj Krishan, and Dr. Raju. I sincere gratitude for coming here. And also our chairman, I extend my thanks to Dr. Navas sir, our chairman and managing trustee to this program, and also extend my thanks to all the senior faculty members and all the participants in today's program. Thank you all.
Thank you, sir.
Let's pay tribute to our nation by reciting national anthem. I request everyone to kindly rise from your seats.
अधिनायक जय हे भारत भाग्य विधाता, पंजाब सिंध गुजरात मराठा द्राविड़।
Thank you.
The bioeconomy was planned to put in, but I took away those slides because we wouldn't waste too much time on it. But we'll briefly look at benefit of genomics and Kerala's contribution to the bioeconomy. And we will already talk about today's rest of the session, so I won't talk much about that. But I think the whole genomics revolution of the last 20 years started with the human genome project. When finally the human genome project became successful. This was in early 2000, but the proper publication of that happened by 2003, 2004. So this was a 13-year long project by more than 11 countries globally, right? So it is one of the largest research projects in biology to sequence the human genome. The human gene structure, general genes, the structure of the genome had already been, the structure of DNA had already been deciphered about 50 years back, right, with Francis Crick and Watson and Rosalind and all that. But nobody still had sequenced the human genome. So the human genome was supposed to be, one of the longest genomes at that time, what we thought. And with those technologies available in the late 90s, you could even sequencing a gene was expensive and it would take a long time. The only technology available was known as Sanger sequencing. So Sanger sequencing, it would take a tremendous amount of time. So the project originally was planned for 15 years with billions of dollars. But luckily, technology progressed during that time, and there were some very competent people, like you see the slightly bald man. He was, what's his name, Amit? I forget Craig Venter. Yeah, Craig Venter was the one who completely changed the whole project. Craig Venter was a junior person. Francis Collins, you see on the other side with the glasses, was the head of the project. He later became the head of NIH. So they were going with that old approach of splitting the whole genome into pieces, having hundreds and hundreds of Sanger sequencing machines in different labs across the world, and sequencing the genome piece by piece, and then later planning to assemble that. That is why he saw the way the power of computing and software and technologies were progressing. So he said, like, hey, that is not the best way to do it. Let us look at a different way. Let us just split the genome into millions of pieces, small DNA pieces, and then it's so easy to sequence them, right? And then assemble it back. So everybody said, like, how will you do that? Genome is just 4 letters. So if you burst that into small pieces of maybe 50 or 100 letters in length, how can you assemble it back? So he said, no, that is why software is there, right? So software can match. There might be some errors, but that's okay. We will get a decent genome much faster than if you go for your old approach. So nobody paid him any attention. So he actually resigned from the project and started his own company, separate company with private funding. And they could quickly move much faster than the multinational projects, the government funded project. So the government and all was so embarrassed. So Clinton and all at that time, who was president of the United States, brought this guy back and forced him to join with the government project. And that's why they both are standing together when the announcement was made. So they all decided to agree to take his approach. So maybe he actually deserves a Nobel Prize. Maybe one day he'll get it. But he's a very aggressive and rude person. So I don't think people like him much. Maybe that is why he has not been considered for a Nobel Prize yet. But he deserves it because he created the revolution of genomic sequencing. And based on that approach, and then newer technologies came also, we could completely revolutionize biology and make it digital. So biology became digital through sequencing. We'll go into that a little more detail, but this was a turning point. So me, a small entrepreneur, I was building a software company at this time, and I was, as Dr. Navas mentioned, California Software. Not a unicorn, but a decent company. We had gone public in India, and I was based in San Francisco, and I was running this company. And our focus, even in California Software, was mostly on source code, in the sense that we would be working with system-level programming. So those who are in IT might know more or exposure to IT, because the software has many levels. You go application level, and which is what most companies do. But we go to system level, where we work with devices. Like, okay, if this device needs to connect to another device, you need a driver. So those type of the programs was the focus of California Software. and based in San Francisco in the Bay Area, what is typically known as the Silicon Valley, there are a lot of companies building products, starting from large companies like Microsoft and Sony and Norton, to small companies coming up with the next new product. You have a phone, new phones and all were starting coming in at that time. You have a new modem. Whatever devices you have, you need a lot of systems level software. So why I'm going that into a little detail is that, so I became very fascinated with the code. You work with the source code. You go down to the assembly level language, you go down to the machine level language, and you need to work with that to get the programs working the way you want. So not at the application level where you don't see the code. If any of you guys might write a program in whatever language that you use now, C or C++ or Java or whatever it is, but you don't see the basic code underneath that. There are compilers that translate the code that you wrote in the software program in English or whatever that language that you use, and it will convert it to the source code. But the computer knows only the source code, the zeros and the ones. So for very complicated level of programming, you actually have to go to the zeros and ones. So anyway, so I was always fascinated with source code. So when I saw the announcement, this genome has been sequenced and you got the source code of life, right? You are able to get life back into 4 letters. Computers had 2 letters, 0 and 1. Biology had 4 letters, AGCT. But finally, all of us sitting here are just a result of that 4 letters code working in each of your cell, right? All of us, anything living in the world, right? Any living thing is made up of genes. So if you are living, if it's alive, it has genes. If it has genes, it has either DNA or RNA. Everything mostly has DNA. Maybe a few things like a virus has RNA only, maybe without DNA. But DNA or RNA. So in our bodies or any organism bodies, the DNA, which is in every cell, which consists of 4 letters. So the letters are, 4 letters mean made of 4 letters, not just 4 letters. The letters can go into billions of letters length, the whole DNA split into chromosomes. So an organism might have 23 chromosomes or 24 chromosomes or 48 chromosomes in each cell. So that is just the DNA split into those chromosomes. And they do the work. They make you, right? So when you are conceived, there is a cell from your dad, there's a cell from your mother mainly, then the cell from your dad, and this fuse together, and you start. And then it takes 9 months and whatever days for the first cell to multiply and become you or me, right? But finally, fundamentally, the base in all of us is the code. So getting access to the code, meaning in theory, we should be able to understand everything about how that organism will develop, how that organism will work, and what can go wrong in that organism. Why is something not working? Or why am I looking handsome, right? And why do I don't have hair? And every little trait is dependent on the genes. Not solely dependent on the genes, but the fundamental factor is the genes. Then there are other factors that affect it. Oh, what did I eat? What did I drink? Have I been using a lot of chemicals to lose my hair? What are the other things in the environment that is affecting it? Even the microbes. So we all talk about the microbiome. Amit will talk to you about the oral microbiome, the microbiome in the gut, which are as many cells as our body. So it's an equivalent organism that is in us. So they also affect significantly. But all of that, what does it affect? They affect our basic genes. The fundamental is the genes that we have for the genetic code that we have, right? So everything in biology now, one of the biggest factors is understanding or taking advantage of the genomics data, right? The digitalization of biology. So I was fascinated when I realized that this is what is going to happen. So in 2009, I started SciGenom. I announced I'm getting out of the CalSoft, and so it took a few years to complete the transaction of selling and getting out. But I could parallelly start SciGenom because I had formally announced that I'm stepping out of CalSoft. So I got a little window when I was in both boards. And SciGenom, I really decided I'm going to focus in India because there are a lot of people playing in the US. Here there was nobody doing genomics in 2008, 2009. So we were the first genomics lab, and I'm proud to say we set it up in Kochi. And everybody laughed at us, right? Hey, how can you put up a cutting-edge lab in Kochi? And so, but why not? Kochi was as good as or better than any other place in India. So we set up it in Kochi. We initially but unfortunately, equipments and reagents, everything you have to import. So we imported the first sequencing machines that came to India. both started off with the Sanger, then other technologies, I'll go into that. My model was very different from normal a startup because for first of all, it was my sort of second innings. So I could think little differently. In the first innings was a software, so I have some experience and I had some money. So I was not dependent on getting other people's funding or any support. I could do on my own. One. Secondly, I felt like, hey, this is an opportunity to do things differently because I was so impressed or fascinated with what genomics could do and the power of genomic sequencing. So I saw the genomic sequencing and the access to genomic code as more like a horizontal, right? In the sense that it is across many fields. It is not one field dependent, right? So it can, the same technology and the same tools that you develop for sequencing the genome and analyzing the data and trying to find out traits that we talked about, hair or whatever linked to genes, that whole process is same for all biology domains. Whether it is a dog, cat, fish, microbe, the fundamentals are just the same. So I felt like this is a great thing to see the technology as a horizontal and build companies in each vertical. Right? So human and medical, this I can make a company. In agriculture, I can make another company. So that was a thought. But I mentioned that to a few people in that time, 2008, and then people would just laugh at me. Right? I mean, what are you trying to do? I mean, how can you do that? So, but luckily I said like I was not dependent on anybody, so I could do the way I wanted. So set up SciGenom. The name SciGenom was just a play on the word genomics. If you notice, it is not the Sai Baba Sai. People could think it is SciGenom. So it is genomics. The last three letters I took to the front. So that's why SciGenom doesn't have an E at the end. SciGenom, and the tagline of the company was Science of the Genome. So very simple. I am going to look at the science of the genome and understand how to figure out the genomic data and its applications, and then see what all the companies I can start applying that in different verticals. That is for different companies, because you can't do agricultural genomics in a medical diagnostics company. You can't do drug development in a healthcare company, right? So you have to have different companies, and you can't do all at one time. You will have to do it one at a time. So that was the plan to do that. But the core technology is the same. So set up a sequencing lab. And put up a dry lab meaning a bioinformatics lab. So typically in genomics it's called a wet lab and a dry lab. So wet lab is where the samples come, whatever biological materials, and getting the DNA or RNA out of that and making the libraries with the right chemicals and putting it into a sequencing machine, and the sequencing machine converts that into the code and spits out the code. And that code then is connected to a good computer from the sequencing machine, and you get the code. Right? So that first part is a wet lab. The second part, when you get the code in, now you have the power, right? So now it is up to because it's billions and billions of data points. So a human genome has 3 billion letters, that can come up to maybe terabytes of data, because of you have to keep in mind that reading each of the letters is very tough, right? The AGCT, the 4 chemicals that you are reading, there's a very combination of high-edge optics. And many other robotics and many other technologies that go into reading the code and then converting it into that. So you can't read one letter alone at once, because that's risky. So typically a letter would be read or probed maybe 30 times or 100 times each letter. So they call it, if you're sequencing, you should immediately ask, okay, what is the coverage? Is it 30x or 100x or 1000x? So your error chances keep going down the more times you read it. Right? But the cost keeps going, and the size of the file also keeps going, or the data that you get. So it can go into terabytes and terabytes of data. So this is really one of the real huge data problems that the whole world faced when you started handling genomic data. But at the same time, so powerful tools in the wet lab can make sense of that, get the right variance that you need to find out what you need to study. Right, so and luckily for me, the whole sequencing technology was went through a rapid change. The turning point was actually 2008-2009. We didn't know that at that time. The next set of technologies were called next generation sequencing technology. Just a simple word that they used, because there's a bunch of technologies. The main company that came out of that was Illumina, so they became the market leader. Even now, about 90-95% of the sequencing, genomic sequencing market is controlled by that one single company in San Diego called Illumina. But they did a dramatic job in bringing the cost of genome sequencing down. So you can see a human genome, even at the time that I started SciGenom, would have cost about $10 million to $8-9 million. Came down to now like less than $1,000. Now for $300 or $400 you can sequence a genome. And what originally took like 10 years or whatever, at this time would have taken like a few months, now can take a day. So today you go, tomorrow 1 or 2 days you can get your whole genome sequenced, right? And at just like 20,000 rupees or whatever, 15,000 rupees. So that is a dramatic price fall that happened. And the other line that you see there, the straight line, the angle line is the Moore's law. So the Moore's law is most of you must have heard of. That is what drove the power of computing technology. So the fall of chip prices and the increase in processing speed of the chip was tracked by Moore's law. So that was so dramatic in the last 30-40 years that we saw the third industrial revolution that I spoke in the morning. So that drove, the Moore's law drove that. So this sequencing power increase was so many times more dramatic than Moore's law, right? So I was very lucky to be at the right place at the right time. Starting off genomics, I had access to the old technologies, new technologies were coming up. India was like completely unexplored land, nobody was doing any sequencing. So I could, we did the first Indian genome in Cochin. Even Malayalam Manorama gave us a 2-page coverage. I was very impressed because normally to get to one paragraph in Malayalam Manorama in Kerala is very difficult. So they were so surprised at what we had done from Kochi, that the whole center spread of Malayalam Manorama was our Indian genome, Malayalam genome. And like normally, like Malayalam Manorama, they gave a good heading to that also. Nammal engana nammalai. I thought that was more impressive than our paper. Anyway, so also we felt that India, we needed to get collaboration, and we need to bring people from outside also to help us. So I also started a foundation, a non-profit foundation called the SciGenom Research Foundation. Dr. Ramchand briefly mentioned to you about the conferences we had. So those are all driven by the SciGenom Research Foundation. Because there is no way that we could do it here. There were no scientists who knew sequencing, there was nobody who knew analytics. So all of that we had to bring from there. But luckily there are so many Indians in well-qualified positions. Some of them helped sitting from there, some of them actually moved down here. So the core team in SciGenom in 2010, I could bring from people who are willing to come back to India. I wanted to look at careers here. Big jump for them, but there are people ready. But there are many other people who didn't want to come, but were willing to help from there, or come once in a year or once in 6 months, and they opened their labs to us. So I could send people from here to go there and spend 3 months or 2 months, even on visit visas, and get trained. And so we quickly became good at genome sequencing. And the first 3 years we focused more on services also. So since nobody was doing sequencing in India, many of the labs were willing to outsource, and most of the sequencing here was going to China or Korea. So we could get that work. Though, like I said, initially there was skepticism that, who can do this in Kochi? So, people from Delhi or Bombay were hesitant to send samples to us, whatever sequencing they had. Sequencing, again, like it can be animals, plants, whatever research projects. No commercial work was being done at that time, but there was always funding from ICAR institutions like CCMB or IISc or many of them. There are many of you may not know, but there are more than 1,000 or 1,200 life sciences institutions across India, and there are more than 100 in Kerala, in the small Kerala. But most of them work in silos. They are not really collaborating. But anyway, they all had some money to sequence at that time. Somebody might want to just sequence 5 genes. Somebody might want to sequence viruses, whatever work. We didn't care. So if you wanted to sequence, send it to us. We'll do it at the lowest price, lesser than what it would cost you to send to China or Korea. And this is right in India. So the turnaround time is much faster. Shipping the samples to other countries takes a lot of. That was a pitch. And people who didn't believe Kochi, I said like, hey, send the samples, we'll do it free. First 5 samples free. You look at the data, if you're happy and it is better than whatever or equivalent to whatever you got from outside, then you can switch to us. So within 2-3 years, we could capture whatever the small market in the whole of India was. Or anybody in sequencing, if they wanted to do sequencing, they would sell it to SciGenom. And that gave me the additional revenue. So I was very particular always in entrepreneurship. Very important thing is that you might have some money, but if you start just use your money, how much bigger the pile is, it will start reducing and you will end up without money. So parallelly, you have to build up revenue too. Especially in complicated businesses like that, it is very difficult to get funding. In the early 2000s, now funding is much more easier in India. But in 2008, 9 and all that, nobody would fund the life sciences companies or science companies. So we would, I had to use my own money and had only limited funds, but so I could build revenue and some margins from that, which was also plowed back in. So 2013, we spun off our first company, MedGenome. So the previous 3 years, we had incubated that, we had built our own tests. So MedGenome focused on genomics research and diagnostics for humans. So medical, MedGenome. So MedGenome became very successful. I'll talk briefly about that in the next slide. Then we spun off, I created another company called SaksinLifeSciences. Dr. Ramchand headed that, and that is for developing a new drug. We followed up that with AgriGenome, so the same concept as MedGenome, but for agriculture, plants and animals, sequencing them, trying to improve them, trying to understand the code and develop solutions. And finally, the last company in that series was MedGenome, which makes kits to actually extract the DNA and RNA out of the organisms. There are kits already available, but MedGenome came up with some new technologies using magnetic nanoparticles, which brings the cost of DNA and RNA extraction very low. And they made a big impact during COVID because they could quickly develop kits for extracting the COVID virus, and they were able to dramatically bring the price down for the COVID testing, as you saw in the second phase of COVID. From a few thousand rupees, the test cost came down to like about 500 rupees and whatever. So one company that played an instrumental role in that was MedGenome. In that second phase, they sold more than about 2.5 crores of kits to people to use it. So those are the things that happened over those years, and by 2020, I stepped out of all these. But I'll just show you a brief growth of MedGenome, because a lot of you are from the medical and pharmacy background, so it might be of interest for you to see how MedGenome grew. So 2011 we started incubating it. Since I knew that I'll be spinning it out, I already gave that the name MedGenome and branded it in MedGenome. We developed some tests to just show the proof of concept, how testing can be done, and started getting revenue. 2013 we raised the first round of funding, $4 million from Emergent Singapore, then put up a new lab in Bangalore and moved it out of SciGenom. In Bangalore, then we invested more on next generation sequencing. I showed you in the graph earlier. So that is the short form for that is NGS. So NGS sequencing with high-end machines. Sequoia, most of you must have heard of Sequoia as the world's maybe most famous VC. So Sequoia invested the next round for us in 15 million, and that was a big boost for us, because getting Sequoia invested in Google, Yahoo, and all that, right? So when you say Sequoia has invested in you, it gives you a lot of credibility. So that was a big credibility boost for us, and Sequoia invested in us. And at that time, we started our lab in US also, not to do diagnostics. So that is mostly for research. So very good advantage that we got was that India had hardly been represented in any genomic studies before, because the Indian government had a rule that the Indian samples could not go out of the country for research. I mean, we all can walk out. We are not actually living samples when we go out, but you can't send a sample per se out of. So even the Human Genome Project of a previous decade, India did not participate. So Indian genomes were hardly available, in genomic data were hardly available for researchers. And Indian genomic data is very powerful because of our lot of history, our ancient population, our large population, our caste system that split up into groups. So we are maybe the biggest genetic experiment the world has ever seen, Indian population. So we have a lot of challenges because of that. Our inherited diseases are huge, but it is very useful data for drug discovery, because there is always a lot of extremities. So there is, if you look at people, I mean, we all follow a normal curve with the tail ends at both sides, right? So with the tail ends are very useful. They are the extremities. So Indian population groups are a lot of people have fall into those extremities. Extreme cases of genetic isolation and foundation of founder effects, all that happens. Anyway, that's a long story, but Indian population data was very useful. So we were able to get a lot of research projects from companies like Genentech and all that. So that funded our growth in revenue. So we could set up a US lab. In fact, Amit, who will speak later here, was the first employee for my MedGenome lab. He quit Genentech, which is one of the largest companies in the US in biotech, but came and joined us. And we were able to do research work in the US, but more focus was in India, and continuing the new test development. So that is where we drove the next set of genomic tests in India, because there was no other sequencing lab, there was no tests available, and we saw great demand for sequencing tests. A lot of children were being born with serious diseases, even in the past, but people had no way to figure out. Now with sequencing, you could figure out, wait, why was this child, what happened? Is it something inherited? Is it because the mother and father are related? Is it a consanguineous marriage? Is that why this kid, or was it just a bad luck? Is a de novo mutation? Typically, de novo mutations are pretty rare, so it has to be most probably inherited. But that doesn't mean that in every case you are able to solve it, the track it. But in maybe at that early hour, chances were like 35 to 40%. We would be able to solve it. We were able to find out like. But still, when I launched MedGenome, I never expected that to pick up. One thing, I was not aware of the depth of the problem. In India, it is more or less hidden. Most of you may not also be aware that how many children are born in India with a serious inherited disease. Because I don't know why, but people seldom talk about it. But about 5 to 6% of children in India are born with a serious inherited disease. I'm not talking about autism or other slower things which happen later in this. I'm talking about serious development disorders like cannot walk, has epilepsy, or neurodevelopmental disorder, or severe disorders like that. So that, if you really look at the Indian births a year, 26 million children are born a year. So 5% translates to more than 1 million children, right? So 1 million children, you can just imagine, are born in India with an inherited disease that can actually be prevented if you know it earlier. Very difficult to do anything about it once the baby is born. So that is why I was talking to Dr. Navas yesterday. So that is all some things that we can really talk about screening and looking at how to apply this. But coming back to my story, so what happened was that this test is all not very popular in the US. So the market, if you look at it, there is not much market at that time. In 2013, 2012, 13, if I look at it, market all in the US is for cancer. So I also started off with the cancer team here, sales, who will go try to sell breast cancer gene testing, But here nobody is picking up because the awareness is very low, and anyway, cancer, oh, you got cancer. I mean, you don't have money and whatever. So who wants to spend another 5,000 or 10,000 rupees on a genetic test? So it's very low pickup. But we started getting a lot of inquiries from pediatricians saying like, hey, can you do this? Then that is a more tougher thing because you are not looking for a specific gene. At that time, even now in cancer, most of the time you're looking for those cancer genes. So testing is much cheaper. But if you don't know what the gene is, and you just know the problem that, okay, child is not talking or can't hear or not walking, it's a bunch of things can be wrong. So you have to do what we typically know as exome sequencing. That means sequencing all the genomes. Not the whole genome, but all the genes. So we do exome sequencing, and at that time it was costly. It cost about $1,000. At that time it was like 70,000 rupees. So 70,000 rupees, who will do was my thought. So we had the test in our list, but we were not pushing it. We were trying to push cancer. Cancer was not selling, but doctors were asking for this test. Okay, we started pushing the exome sequencing. A lot of people are sequencing. And we were thinking like, why are they doing this? Why would a parent spend 80,000 rupees for a baby already born with a disease? And everybody knows gene disease, I mean, you only in the future maybe gene editing will come, but till gene editing comes, you can't do much about it. Maybe in a few cases, if it was an enzyme deficiency or whatever, you can supply, you can give something, but even by the time you diagnose the baby, would have been damaged, like in first 2-3 years of life without getting the proper enzyme. So can anybody guess why people still were very desperate to do the sequencing? Yeah, madam knows. Next baby. Right? So that was the biggest driving factor among all families. So if you had a baby, then unless you're very illiterate, then like I know there were families with like 7 children and all, everybody had a disease. Right? So that came as a big news and all that. But otherwise, people would know that, oh, no, it is not because I didn't pray or I was a bad person or anything. There's something else that happened, that this happened. So, and there is a science now to at least find out what is wrong. And then I can maybe try to see how we can avoid it for our next baby. So that is why that really drove the demand. Even now for MedGenome, the company has grown tremendously over the last 10 years. I mean, I'm no longer in the company. I stepped out just during the COVID time. But it is the inherited disease testing of pediatric children still continues to be the largest revenue segment. So, but again, so I'm sad about it. I'm not happy about it. I'm sad about it because that is done after the baby is born. At the same time, MedGenome and other companies have screening tests by which couples can do the screening before either they marry or before they have the child. But how much ever we try to market it, very few people do it. Only when they get hit. It is true that the chances are only 5%, but 5% is not a small percentage when you're talking about a baby. And so, it costs only less than 15,000 rupees for 2 people, so about 7,000 rupees per person to do test. Somewhat expensive, but there's no way we could bring the cost below that. I mean, because it's the way we import the machines, import the consumables, there's a minimum price that we can offer it at. But very few people do till a baby is born. Then they do all these tests and find out, and there are various ways to try to prevent it for the next baby. So anyway, coming to is that, so awareness and other measures that we need to do can drastically reduce this impact on society. Because just think about the baby, think about the family, think about the society in large, think about the total cost of one baby being born with a serious disease is huge. And we have been very successful in eradicating diseases like polio and things like that. So there is no reason why we can't take steps to practically eradicate about 1,500 diseases, inherited diseases that are common in India or more prevalent in India. Anyway, so the MedGenome has done well. We launched a number of tests in India like this first-time test. NIPT is a very non-invasive prenatal testing. Is anybody familiar with that? NIPT? Yeah. So NIPT is where you just take a mother's blood and you can look for many of the diseases. Not all like the panel that I talked about earlier, carrier screening, but many of the chromosomal abnormalities like Down syndrome and all that, and about 8 to 10 diseases like that can be found out. The decision is tough if the baby has that, because the only decision is to abort. So the test has to be done legally before the 20 weeks or 22 weeks. And it's a tough decision, but at the same time, it is a decision I think most people would take than to have a baby. And again, people might think the chances are low. In fact, I had very tough experience with Dr. Devi Shetty. You might have heard of Dr. Devi Shetty. Devi Shetty is one of the most well-known cardiologists in India. He runs Narayana Hridayalaya Hospital. He is a revolutionary leader in India who brought the cost of cardiac surgery and all to a very low, affordable person. So I happened to visit him at that 2015 time frame when I was setting up the lab in Bangalore, MedGenome and all that, and we had launched NIPT test first time in India, NIPT knowledge. And he was actually very angry at me, and I was like, he's saying like, you're doing such a poor job marketing. And I said like, no, we are trying our best. We are trying to tell people, doctors, convincing them that all pregnant ladies should do the NIPT test. It is very simple. Just draw the blood. There is not an invasive test. So it's not like a amniocentesis or other typical tests that you do. But again, India is very difficult. There are no protocols. So if there were protocols, you could change protocols. But if there is no protocols, what do you do? So every clinician you have to convert one by one. India is maybe one of the only countries in the world I have seen where there are no standards. So everybody any hospital can decide whatever protocol, whatever standard they are going for. So why doctor was unhappy or angry with me was that he said not that we were not selling enough. Then he felt that I was not getting fully convinced. So he dragged me into his neonatal room, ICU room. It was like, you feel so sad. 50, 60, 70 small babies, all with heart problems, lined up like in a big hall, right? So he's saying like, see, all of this could have been prevented if the parents had done the NIPT. So people get very emotional and in the field who are handling that, and very sad that this is not being adopted. While even countries which are, I would say, in a very different mode like China just made a rule in 2016 or 17. Every woman pregnant will do NIPT. I'm not advocating China in India because we don't want it to be China type of government, but even I'm saying they did it, and so they dramatically reduced these type of babies born with these type of defects. So it can be done. Anyway, so like I said, I finished this innings on SciGenom, and I stepped out of all the companies. I still kept my core lab, SciGenom lab, and the foundation, SciGenom Research Foundation. And SciGenom Research Foundation has always, in the last 10 years, been focusing on research and education. The conferences that we mentioned, we really focus on some publications. So as you know, it's so